Good morning. It's my privilege to present on behalf of the co-authors the second half of this poster discussion concerning mechanisms of resistance and response to endocrine CDK4/6 inhibitor combinations. In today's block, we're really looking at the question of how intrinsic subtype might influence the outcomes of patients on these therapies. First, these are my disclosures. These are also available at the ASCO website. So we know that genotype as well as intrinsic subtype can influence tumor response and outcomes of patients on endocrine therapies and endocrine CDK4/6 inhibitor combinations. Shown here are two examples of that. First, work from our group recently published that shows how genotype can influence response, that several genetic alterations are associated with short response mechanistically due to loss of RB or to upregulation of CDK6 kinase, and these were then validated in the laboratory. Secondly, recently published work from Alish Pratt, the first poster we're going to be discussing, where they looked at the impact of intrinsic subtype on outcomes on the Mona Lisa trial, showing that the basal subtype had a poor prognosis and outcome on these combinations. And one would really envision integrating this information, genotype together with intrinsic subtype, as potentially giving us really granular information that might tell us about outcomes. This is just an example of how integration can really be powerful. This work from Christina Curtis looking at outcomes of breast cancer patients using integrated clusters along with IHC subtypes. So today's session really covers three posters that look at this question in different ways. First, looking at integration of genotype by circulating tumor DNA together with intrinsic subtype by PAM50. Secondly, looking at the method for intrinsic subtyping and how that might be influential. And third, looking at changes in intrinsic subtyping and how that might influence outcomes. So first, from Alish Pratt and colleagues, looking at genomic profiling by circulating tumor DNA across intrinsic subtypes on the Mona Lisa studies. In this study, intrinsic subtyping was done on both primary and metastatic tumor samples where available, and circulating tumor DNA was analyzed at baseline upon entry into the study. And the focus here was really on genes that were altered in upwards of 5% of patients. And shown here is the sort of broad overview of the data. You can see different intrinsic subtypes and then the occupants of the genotypes found by circulating tumor DNA. And some of the expected findings, like PIK3CA being the most commonly mutated and enriched in all of the groups, but highest in, but a little bit lower perhaps in basal, P53 being dominant in basal, and overall gene amplifications being more frequent in all the subtypes than luminal A. I will point out a somewhat high rate of DNMT3A alteration seen in the luminal A and B population, which does make one wonder if there is some influence of clonal hematopoiesis in these analyses, given that this is a much higher rate than is seen in other somatic tumor profiling studies. Nevertheless, several genetic alterations could be looked at and to understand where they fall in terms of the different subtypes. So again, amplifications like cyclin D1, FGFR1, MYC, and other alterations enriched outside of luminal A. And ERB2 in particular, in this case mostly missense mutations, are less frequently seen in the basal subtype. The circulating tumor day fraction itself different between the groups, highest in HER2 and luminal B compared to luminal A. The basal-like, just not as many cases, so while numerically higher, not necessarily statistically significant. So what are the key findings overall? I think it's clear that there are differences in the genotypes present in these different intrinsic subtypes, and that could influence and enable some of the sort of integrative analysis and influence outcomes. It's clear that all the groups benefit from the addition of CDK4/6 inhibitor to hormone therapy, albeit with the basal-like small population potentially not, although that, again, a very small group of patients. So onto the second study, this being led by Maggie Chang at the ICR, really looking at the question of how do we call intrinsic subtypes, in this case using two clinical trials, the Paloma-2 and Palette trials that looked at the addition of palbociclib to letrozole in the metastatic or adjuvant setting. So by way of reminder, the PAM50 algorithm, which is known as commercially as Prosigna, is used to classify tumors into intrinsic subtypes. And there have been computational methods to convert different gene expression methods into a PAM50-based intrinsic subtyping with some efficacy, and this is increasingly being utilized for different gene expression data sets. In this study, these were compared, and so both in the Paloma-2 and Palette studies, aims done by using EdgeSeq expression data was compared with conventional Prosigna PAM50 classification. And really, the punchline here, you can see important differences in comparing Prosigna versus Ames-based classification. And highlighted here, 46% of luminal subtype called by Prosigna were assigned luminal A by Ames, and 67% of the basal-like that were called by PAM50 Prosigna were assigned HER2-enriched by HEG Ames. So important differences, ones that potentially could impact therapeutic decision-making in the future. Now, the actual outcomes of patients, in this case using the Prosigna study, do show, of course, the benefit of the addition of palbociclib to letrozole across the subgroups, with the exception, again, of the basal-like, where there are significantly less patients. So overall, of course, palbociclib is proven to be effective across the subtypes, with the possible exception of the poor prognosis basal-like subtype. But importantly, a standardized PAM50 assay, at least now, should be used until such discrepancies can be worked out. Finally, this poster led by Carsten Denkart and colleagues, looking at, in this case, a possible change in intrinsic subtype, pulling data from the Penelope-B trial that looked at the benefit of adjuvant addition of a CDK4/6 inhibitor, palbociclib. So in this analysis, they utilized, in this case, the Ames-based subtyping with samples collected pre-treatment and then post chemotherapy, neoadjuvant chemotherapy. So the sample collected at surgery. And they ran essentially a comparison of these and asked whether either of these in their comparison might influence outcomes. So they had 540 patients for whom they had these paired pre-treatment and post-treatment samples. And so shown here, of course, the prognostic effect of subtyping was preserved, whether you looked at a pre-treatment analysis or a post-treatment surgical analysis. You can see, of course, again, basal subtype doing poorly, the relatively good prognosis of the luminal A and B. But what was really striking was the change within patients from one subtype to the other, from the pre-treatment to the post-treatment. And so that's really depicted here, where you can see, of course, the samples that stayed the same versus those that changed. And when you asked what happened in terms of outcomes for these groups, you can see that, of course, here, luminal Bs, no benefit for the addition of palbociclib here in brown. Same for these pre-treatment analyzed luminal As, and same for these luminal As that were preserved here. But this group of luminal Bs, which really seem to preserve the luminal B status, really seem to benefit from the addition of palbociclib, albeit with a small number of patients. So what does this mean? First of all, what does it mean that there was a change? Was there actually a change in the biology of the cancer cells as they moved through chemotherapy? Or rather, was there a selection for chemosensitive versus insensitive populations? And does this imply that a failure of selection for chemo-insensitive populations, that is a lack of chemo response, perhaps predicts for the subsequent benefit of the addition of a cell cycle inhibitor? Much more to be gleaned and learned about the biology here. For instance, what are the genotypes between these populations? What is the basis for sensitivity or resistance to the addition of a CDK4 inhibitor? But really fascinating to see this change going on. So in closing, I just want to emphasize that this data, really exciting. And now the next questions that emerge are, how can we use this information, perhaps integrating with genotypes that we know don't respond to CDK4/6 inhibitors? We know that patients can do very different. Some progress right away, some stay on therapy for a very long time. How can we use this information to ultimately make predictive changes? Should basal-like tumors be randomized to this versus a different targeted therapy or even chemotherapies? Such questions, I think, are now ripe for investigation so that we can convert this information to something that is clinically actionable. So with that, I'll close by inviting the investigators to be part of the Q&A session next. And I thank you for your attention.